I like to talk about the history of PFA because when I first started working in this in 2011, the way I started was looking up every way we ablate soft tissue. Every way, ultrasound, lasers, diphtheria toxin, alcohol, x-rays, like all the ways you can do it. And I listed the pros and cons, and then I kind of walked down the line. When did they start doing this? Why did they do this? Why did they give it up? And that's how, we kind of started, or at least I started. And at the time, I did not realize how many other people were looking at the same thing. It wasn't just me. It was Ashwattham at Mayo. It was Whit Kempf and Karsneven in Netherlands, yes. And the guys, Medtronic and Abbott and Johnson & Johnson all had groups looking at it. PFA, I don't know about the guy on the right, but the PFA was coined. It's a marketing term, and by a marketing genius named Mark Stewart, who is an employee and engineer at Medtronic. So we call it PFA, but guess what? You can call it fulguration, you can call it DC ablation, you can call it electroporation, you can call it whatever you like. It's really electric field mediated tissue injury, where the primary mechanism of tissue injury is caused by exposing it to an exogenous electric field that is great enough to disrupt the metabolism of the cell to such a degree that it cannot survive. But the first electroporation was really a bunch of Neanderthals walking around on the plains and getting hit by lightning. And if you get hit by lightning, it's a very strong electric field. And not only might you get a burn, there's actually been proof that these Lichtenberg figures are largely electroporation responses. You can electroporate the cell. And there's actually some interesting theories out there of evolution relying on lightning strikes to open up, the membranes of bacteria in order to facilitate exchange of genetic material. So, I looked at the history of electric field mediated tissue injury, and I went all the way back to 1886. And then I kind of walked around to find out where did it start and how did it move forward. The first thing I could find was 1873 in the British Medical Journal. There was one thing in 1867 that I thought about it. Galvani and Volta and all these guys were working on bioelectricity and identifying the ability to make, capacitors, Leyden jars, and placing it on everything. And eventually the excitement of this kind of technological revelation and revolution led to generators that could create a lot of voltage, static charges and continuous charges, that people were just curious and they would start putting probes everywhere. And there's a whole bunch of publications from the late 1800s to the early 1900s where people were doing tissue ablation using very high voltage pulses. And that's where the term fulguration kind of came from, because one of the ways that people did that is by sparking into the tissue. And so there's some interesting descriptions that took place. Fulguration, that term again was used, and it was primarily used and most effectively in cancer treatment, where they were sparking into usually dermal lesions like basal cell carcinoma. Here's an example of a soldier in World War I getting treated by one of these devices. In fact, Tesla actually has a patent on a fulguration device meant for medical purposes. And so people were aware that electricity could damage tissue. How did it work? We don't know. This guy Turner on the bottom, 1919, California Medical Journal, 1919. This is a comparative study where he takes diathermy and compares it to fulguration in patients who have basal cell carcinoma. He's firing sparks into one, and then he takes the probe and he puts it over the other one and he cooks it. Diathermy is basically a form of radio frequency, a little bit lower frequency rate, but it cooks the tissue. He followed the patients, took punch biopsies, and described it in this publication. This is like one of his only publications. And then after that, high electric field mediated tissue injury sort of goes away. What he found was that the cancer regressed more with the fulguration than with diathermy. But the patients liked the diathermy because they felt like they were getting their money's worth because the doctor, they was cooking and there's fire. They felt like they were doing something. Because with fulguration, the lesion looked exactly like it did when they walked into the doctor's office. But there was something happening inside those cells. Fast forward to 1975, and I will point out that there is so much science ongoing in these days to identify cell membranes, electrical responses of the membranes, capacitance of membranes. So all this membrane knowledge was kind of growing by lipid layers, et cetera, how proteins worked, how they were. And then in the 1970s, a couple of really smart guys figured out, hey, we could use strong electric fields for microbiology to help transfect DNA and do a whole bunch of stuff. So people were looking at it. Electroporation was kind of coined by Newman in the early 80s, but he was really doing his work in the 70s. And then in 1975, on a completely different level, a different guy, James Bessel, not only invents PFA, i.e. Electric field mediated tissue injury inside the heart, he at the same time also invents catheter ablation. Prior to this, anytime you were an electrophysiologist, we would have all these catheters, we'd put them up in your heart, we'd pace, we'd look at the conduction velocities, we'd give you a drug. We'd saturate the drug and see if it stopped the problem. If it didn't work, we'd keep you in the hospital, change the drug, bring you back, pace, diagnostic stuff. But if you needed an ablation, you would have to have open heart surgery. The first time anyone's decided, and off to the right here, this guy was a physiologist working for Pacesetter, and he decided, hey, I'm trying to figure out this animal model, and I have to do open heart surgery, 3 hours of surgery every day, and that's a lot of work. And he decided, I'm just lazy, so I'm going to take this Brocken-Brown needle, put shrink wrap over the top of it, create a catheter, put it up against the AV node, and attach it to a defibrillator. And when he did that, boom, first time he did it, it worked. He had the model, and he went forward. Yesterday, we honored James. He couldn't be here tonight. A lot of people don't recognize this guy because he's not a doctor, but Mel Steinman knew this guy. And Mel Steinman said, hey, maybe we could do that in people. In between James and Mel Steinman was a guy named Guy Fontaine. He's a French guy sitting there doing the EP work, and there was an error in his defibrillator, and somehow the defibrillator patch got attached to the wires going into the patient. So I think this is a device deficiency and a complication, but needless to say, the patient ended up with heart block after the ablation, and he might have been the guy to do the first DC ablation ever. He published on that later, but the first guy to actually systematically do it in a patient series was Mel Steinman. He's a giant in the field, and Gallagher, who he was at Duke. They both published a series. Now Gallagher said he came up with it on his own. Mel Steinman said he was working with Bessel, and they both published on this new technique of taking a defibrillator and attaching it to a catheter and moving forward. Oh, sorry about the music is there to set the mood. But what DC ablation is, you create a very strong electric field, and in this case, in saline. In the old days, those catheters basically would allow you to have very high current densities, and electrons, despite finite element modeling, do not move from your anode to your cathode in bodies. It is not metallic electron motion. What it is it's saltatory motion. It is electrochemical motion. The electrons are moving through electrochemical reactions causing electrolysis, or you're causing concentration gradients of charged moieties, i.e. a battery. That creates gas. The gas converts into plasma. Plasma has more space and it pushes out. And so the early days of DC ablation, everyone was worried about this phenomenon called flash arcing and whether it was causing trauma. And the main mechanism of injury initially was, we think the ablations are working via barotrauma. And then a number of very smart guys in England, Cunningham and Rowland at the London Heart Hospital, tried to solve that problem because there were some complications at the time. So they reduced the pulse duration. And they built the first commercially available PFA system ever. 10 microseconds, 5,000 volts, multiple outputs, synchronized. And so just go back to the 1980s and you will find that everything we think is new now, other people thought of already. Another really smart guy who's still around and doing really big things is Gus Barty. So this guy, he was a young man at the time, but he was looking at, is it peak voltage better than total energy? And he did this very elegant study where he was able to take the same exact amount of joules of energy and either separate it with some high voltage pulses or take a lot of little tiny voltages on the bottom. He applied them to this animal model and he looked at them on day 1, on day 30, and looked at the histology. And interestingly enough, same amount of energy, but you get bigger lesions with higher peak voltage. And so it was a clue that it's an electric field mediated tissue injury. We got some very smart guys here, Boris Rubinsky and Rafael Davalos. Davalos might be around. I hear he's in the walking around HRS. These guys kind of reinvented DC ablation, but with a more modern pulse generator. It was called the NanoKnife at the time. Super smart guys. But they worked with this guy, Levy, and they took a left atrial appendage clamp or a pulmonary vein isolation clamp and attached it to a pulse generator and caused ablation. And they showed that data. And this is this. And so even though between 1989 and 2007, I think is when they published this, there were doctors still doing good old-fashioned DC ablation. Steve Bayless, a number of other people that I know actually have published on this. And so it had never fully disappeared. But here we go where people are bringing something new and it's ignored. Another group, Whitcamp is working in the Cars Nevin. This is his group. They started looking at all the ways and the potential advantages. Ashwattham at Mayo Clinic was looking at this. This is 2013, 2012. I'm working on this at the time. And so there's a lot of data kind of building up. And so this is the first catheter that I built for Farapulse. It was an epicardial procedure at the time. My idea was that you want to avoid strokes, go epicardial, wrap it around the pulmonary veins in the posterior wall, and then fire off this 30 electrode catheter. I didn't have any money. I had to build those myself, but those worked. Actually, I see a guy over here who helped build them. Eric's over here. This is one of the first animal studies with the first IO-Approach / Farapulse generator. And the problem with high voltages is you get capture of tissue. Anything that's excitable, you can capture. We call this the rodeo. But I looked very calm. Like, it doesn't even faze me. So Farapulse kind of, the only thing, since so many people knew about this, it had a rich history. The history of PFA is mostly marketing and all this stuff that's going on. But I think what changed here was I was dumb enough to start a company as a physician and run with it. And Farapulse took 8 years to get it over the finish line, but it's doing a great job out there. And so I feel like we've solved the problem in the atria. Now it's time to go for the ventricle. And boy, there's a lot of companies out there competing for that pulmonary vein isolation. Every shape, every color, every kind of thing. Thank you, guys.